Hello, guys, and welcome back to the second part of chapter 14, which chapter 12, patterns of inheritance. So, as we saw, we spoke a lot about Mendel's kind of genetics, and we learned a lot about his law, his basically his kind of model, you know. And interestingly, this is an interesting fact for most of you guys. During Mendel's, basically, he published all of his works and everything, but it never got actually so much hype. He expected people to be very excited for it with his work, but not really. It was only like actually like 30 years later or something that other scientists had, observed exactly what he saw, and they wanted to publish their work, but then they found his work. They're like, oh no, crap. We actually are just repeating what Mendel actually brought into light. And that's when his work started to become very interesting during those time. During his time, it never really got actually so much hype. It was only later on with other scientists also starting to see very similar stuff to him. So that it actually got really kind of hyped during those times. They're like, oh wow, we can actually use now we understand how these traits are actually inherited. So because it got so much hype, so many scientists were trying to actually investigate this, but they were not finding the same ratios, these very simple ratios that he was expressing this, 1 to 2 to 1 or 9 3 3 1 that we spoke about a lot. They couldn't find it. And it made a lot of sense because Mendel was using a very simple um, model to study genetics, which was the peas. The peas are very simple because their characteristics and their genes are very simple. It assumes if you take their genes and they're basically their whole pattern of inheritance, it basically assumes that each trait is controlled by a single gene. So the color of the flower is controlled by one gene. The color of the pea is actually controlled by a gene. If it's smooth or wrinkled, the pea is by one gene. And each gene has basically two alleles, meaning it has um, two alternative traits. So the gene, for example, in color has two alleles. Either it could be basically purple or white. If it's basically the shape of the pea, it's either round or wrinkled. So you only have two alleles. And that there was a clear dominant recessive relationship, meaning that if one allele is dominant, it basically is only expressed while the recessive one is not. So it was clear. However, this is very premature because this might work with peas, but it doesn't work with, let's say, more complex organisms like, you know, certain animals like pigs or cows or us humans. It's much, much more actually more complex than you think. And we're going to actually give you examples in where these things do not apply at all. So the first one is polygenic inheritance. So polygenic means, as the name says, poly is a lot, genic is, genes, meaning that one trait can be actually governed by multiple genes. So it occurs when multiple genes are involved in controlling a phenotype of a trait. So the phenotype is an accumulation of contributions by multiple genes. So meaning that one phenotype is not just the product of one gene, but it's a product of multiple different genes. And this gives you something called continuous variation, which we're gonna actually, go more into details about that. But I want to use an example here to actually make this easier, which is human height. How tall are you? Which is something very, people really take into consideration a lot. It's very important for people to be either smart, so to called short or tall. So the human height is controlled not by one gene, it's controlled by multiple genes. So it's the product of these multiple genes that tell you your height. Because imagine if this was actually controlled by, one gene, you're either tall or you're small. That's basically what it is. We would be having two categories. People are just purely one small and one height small and one height that's tall. That's it. But we don't see this. And basically we see this very nice kind of distribution, right? People, having a height of 5 feet going to 6'2", basically, in height. So you see this distribution, and this is what we mean this continuous variation. This continuous variation happens because it's not controlled by one gene, it's multiple genes. So it's basically combinations. So that's why we have this basically distribution or this continuous variation that we have. And that's, the simplicity of it. Because if, genes, if basically the height was controlled by one gene, you literally would have here people high, like basically this small, and people this high, and that's it. You wouldn't have anything in the middle. But because there's multiple genes, it's variations that come out of this, which is very cool. And actually, you see a lot of new things coming up. It's not just boring. Either you're short or you're tall. No, you have a nice, variation among. Then you have pleiotropy. Pleiotropy is basically very similar to polygenic inheritance, but here we're changing a bit the sequence, where actually the phenotype, so basically an allele has more than one effect. So here is that the gene is basically, if a certain gene is actually mutated, it actually can have multiple effects in the phenotype. Not, you don't have just one phenotype. So here, basically, gene that affects one trait often performs other unknown functions, meaning that when that gene is mutated, you think that it will give you just one phenotype, but no, it gives you multiple phenotypes. And I'll give you an example here with two actually diseases, cystic fibrosis and sickle cell anemia. So cystic fibrosis is a hereditary disorder. We spoke about this actually. It's a disorder that actually you get a mutation in one of its actually chloride ion channels. So it's not able to actually, when you have this allele, your ion channels, your chloride ion channels in your lungs, they do not work. They're actually not folded properly at all, and they cannot work at all. So what happens is that you cannot remove chloride ions, and that actually is a huge issue because then you cannot remove mucus in your lung. So you think that's where it stops. Is basically you just get mucus in your lungs. No, just this one gene is causing so many different types of actually phenotypes like liver degeneration and lung failure. We went from just one phenotype, which is basically abnormal gland secretion, so thicker normal mucus, into straight away liver degeneration and lung failure. So we saw how one gene can be mutated and give you multiple types of phenotypes. That's pleiotropy. Then you have sickle cell anemia, which most probably you guys have heard. It's a certain mutation again, and this one actually affects just how your red blood cells are. So normally red blood cells are actually quite nice and round. Well, when they get actually this mutation, which is in one gene, this whole red blood cell actually becomes more curvy. So actually, its name comes from sickle cell. It actually looks like a sickle, like a very like kind of the shape is quite weird. It's not around anymore, and these guys start to bind together. So this is the phenotype, right? This is one phenotype. But no, you actually get a lot of different problems rising from heart failures, rising to lung problems, to also issues when it comes to actually bleeding. So multiple types of phenotypes just from one gene. So this is what I mean by pleiotropy, that not every genotype gives you one phenotype. So what meaning that not every one gene gives you one phenotype. No, it can give you multiple actually phenotypes, just one gene. And then we also thought that, for every gene, you actually had only two alleles, so two basically, variants, two variations. Like you have the gene for the color of the flower of the peas, you only have two alleles because you either have, white or purple. In humans, that doesn't exist. And the most clearest example, which I'm quite sure you know, is the ABO blood types. In humans, we have either type A, type B, or type O, and that's because we have three different alleles. So when it comes for the gene of like what is your blood type, you can have three different alleles. Either you have the A blood type, the B blood type, or the O blood type. So that's what I mean. And this fourth thing that the relationship between dominant and recessive is quite clear, meaning that if you have the dominant allele, it's going to express, and if you have the recessive allele, it's not going to express. Well, that's not true. And we're going to show you here two examples, incomplete dominance and co-dominance. So with incomplete dominance, what it means is that the heterozygote intermediate in phenotype is the basically is the two homozygous. So meaning that, for example, if you actually take the red flower and you actually give it to the basically cross it with a white flower, you don't get either red or white, meaning the red being actually the what's it called, the fully dominant, and the white being fully recessive. No, you get intermediate because you have both genes and both genes are being expressed. So most probably the red is being expressed more because it's a bit more dominant, but it's not a complete dominance. It's incomplete because the white is still being able to express, but not fully. So that's incomplete dominance. And then you have codominance, meaning that the heterozygote you get. shows an aspect of the phenotypes of both homozygous. So here you see both being expressed equally. And we will take an example here with AB blood type and actually the basically the cattle. But we're gonna just take the example of AB blood type in a bit. So let's go a bit more into the incomplete dominance. So we have basically, in this case, a red flower that we know is homozygous. So it has basically the genes for the red, so I've seen here big capital R's, and that's red. And then we have the white, and this one is basically dominant but not super dominant. It's basically in the middle. So when you fertilize them and you have the F1 generation, you would get this purple flower. You only get purple flower. Remember before when we actually crossed the purple peas and the white peas, you only got purple peas because there was a pure complete dominance over purple. So purple had was completely dominant over the white. Here, not really. When you basically have now a flower that has the allele of the red and the white, you have this incomplete dominance. So the red is mostly more dominant, but it's not 100%. There is some incompleteness. And if you take now this basically uh, kind of pink flower and you self-cross it, so this is what you get if you put it in a Punnett square, you will get this nice, the same exact ratio, this 1 to 2 to 1. So two purple, sorry, two pink, one red, and one white, basically. Because you still actually have, and here in this case, you still actually have the same ratios when it comes to incomplete dominance, but the phenotypes are a bit different. So rather than having three um, red, you have actually one red and two pink. So let's talk about now more the human ABO blood groups. So this is an example of actually co-dominance. So where both actually genes are both dominant together. So when it comes to actually the ABO blood groups, we know that there's multiple alleles. So it's not two, it's actually three alleles, which is in the I gene. So the I gene is the one that actually gives you the blood type, and there's three variations, or three alleles. IA, which is basically the one that gives you actually blood group A, IB, which gives you blood group B, and this I lower, because this is recessive, that gives you O. And we know that A and B are dominant, and they're basically, they're dominant over I, but they're co-dominant with each other. Meaning that if you have AI with this small I, AI is way much more dominant, pure dominant. That AI is not gonna actually be expressed. While if you have IB with I, also it's the same story. This is super dominant. This is gonna be expressed, and this is not. But if you put these together, IA and IB, they're co-dominant with each other. Both of them will express. That's what we mean. And as we know that the blood basically what determines group A, group B, group O, very simple. It's just the sugars that is exhibited. So what sugars do you have in your surface proteins in your red blood cells? So if you have, for example, these basically blood type A, you either are poly IAA or I, so basically having basically you're homozygous to the allele of A, or you're heterozygous but with a I, because we know that IA is dominant over the small I. So if you're blood type A, you either have either of these basically copy of genes. And basically the sugars that you actually have exhibited in your red blood cell is galactosamine. But then if you look at basically blood type B, it's very similar. You either have basically homozygous with B alleles or heterozygous with the B allele and the small I. And you basically have galactose. But when you have basically both alleles A and B, you're basically you call them blood type A and B because suddenly now your blood cells have both galactose and galactosamine. So both of these. So both are being expressed. So this is where the codominance factor comes into place. That there's no clear relationship that dominant is this allele and basically recessive is this actually allele. No, in this case, both of them are dominant and both of them will be expressed equally. And then, of course, we know that I is recessive. So if you want to have a blood type, basically O, you have to have both the recessive alleles together. So basically, you have to be homozygous to the O allele. And last, basically, one more other thing that you also want to take into consideration is that Mendel also said basically he did something a bit naive that he said, oh, genetics, is just being passed on, and that environment really doesn't play an effect. Maybe with the peas because it was in a very actually kind of secure environment where the environment didn't change. But we are humans and animals also, for example, we live in environments that keep changing on a constant and on a daily basis. So the environment can actually have a massive, huge actually influence in your genetics. It's crazy. Trust me, it's crazy how much environment can actually have an influence. And I'm going to give you here a very nice example with basically this Siamese cat. So Siamese cat have this really nice kind of phenotype, and where they have their heads is more kind of, let's say, dark, and especially their tails, but their main body is white. What gives that really beautiful phenotype is very simple. So basically, there is this really nice enzyme that's called tyrosine, and this tyrosine, what it does, it basically creates more, or basically kind of starts to produce more pigments. So if you have more pigments, you're going to become a bit more black, basically blacker. So in the face and in the tail, basically the temperature is below 33 degrees. So the actually that protein is actually working much better because it doesn't like to for the temperature to be higher than 33. So it works more better. So you start having more pigments. That's why it's a bit more basically your the face of the Siamese cat is darker and basically the tail also. But then the main body, as you can see here, basically the temperatures are above 33 degrees, and the tyrannase is inactive. It doesn't like it. It doesn't do work, and it doesn't do create pigments. So you get the color white because there's no pigments. There's no black anymore. So white comes out. So this is the cool thing how environment can influence it. This is a very simple actually example, but trust me, it's quite impressive how much the environment can influence this. And the last thing I'm going to talk is about epistasis. So epistasis might be a bit complex, but it's not. So epistasis here actually ruins a lot this whole thing that Mendel was saying about, if you guys remember, this the independent assortment. And where he said where two basically genes are not affecting each other. That's what he said. Well, that's not true at all. And that's epistasis. Epistasis says that the interaction of genes that are not alleles, so meaning that these genes are different, they're from two different, basically, giving you two different traits, can actually affect each other. So one of them can suppress or one can affect the other. And how can they affect each other? There's four ways. Either one gene affects the phenotype of another gene due to the interaction of their gene products. So one gene is basically, so bearing in mind, always think about this. Genes are basically just big pieces of or big recipes for proteins. They encode for proteins. So one gene, when it basically gets translated and transcribed, which we will learn more about it, it's going to be turned into a protein. This protein can actually go affect the protein of another gene, and this can really affect the genes actually expressions or actually alter their phenotypes. And we'll definitely take into more actually this year. We'll actually talk about it more. I'll give you more an example of that. So the expression of one gene in one locus, so locus means in one location, can alter the expression of a gene at a second locus. So one gene that's completely in a different location can actually, for some reason, really affect the expression of another locus. This is a bit more complex, but just what I want you to know is that basically, if one gene is a bit far away, its expression can still actually affect the location of another gene that's far away. Second, two genes encode proteins that participate in a sequence in a biochemical pathway. So, guys, remember biochemical pathways? Biochemical pathways are reactions that happen, or basically a series of reactions that happen one after the other. And as we know, there's a lot of enzymes that regulate this whole biochemical pathway. And it could be that two genes are actually participating in the same biochemical pathway. So the two genes are encoding for enzymes that work in the same biochemical pathway. So they actually affect each other. And we'll give you another example of this one, actually. And one gene can mask the expression of the other. So one gene, the product, can actually mask the product of the other gene. So one gene, so let's imagine gene A. Gene A gives you protein A. And then you have gene B that gives you protein B. Protein A can go and actually, hide or kind of stop all the work of protein B. So it stops all the phenotype there. So that's how epistasis can occur. And we're gonna give you here some examples. And I'll give you the first example here is with corn. And this example is where, how two genes encode for proteins that participate in the sequence in a biochemical pathway. So as we said, the biochemical pathway is the series of reactions that happen one after the other, and basically there's multiple enzymes involved. And R.A. Emerson found out that when you had two varieties of corn, and when he mixed them, so he knew that basically these corns are either white or they're either purple, and he knew that it was actually controlled by two genes. So when he crossed them, he thought you were gonna get this 9331 ratio, the one that you got, that Mendel got when he actually crossed two different basically genes. Remember the basically the round, smooth, and basically yellow peas with the wrinkled green peas. He got a 93:1 ratio. So he thought he was gonna get the same thing here, but no. He actually got nine purple and seven white. So what happens here is that in these actually corns, the allele for either product, a gene that produces a non-functional enzyme will result in a lack of pigment. So these non-functional alleles are recessive to the functional alleles, so the presence of at least one functional allele. So here, okay, so let me go here. So basically, this whole basically you have white corns or purple corn. So you have here this basically white corn that basically is controlled by two genes, as you can see here, the AA or BB, basically. And these basically the AA or BB actually are producing genes or are producing sorry proteins that are working in a biochemical pathway. And you need both of them to be functional, because if one is basically completely unfunctional, basically then once the basically the whole biochemical pathway is completely actually stopped. You need basically these products. So here it says so that an allele for either gene that produces a non-functional enzyme will result in a lack of pigment. So the pigment that gives the purple color is basically going through a whole biochemical pathway, and you need all of the enzymes. If any of the genes are giving you a doesn't give you a functional enzyme, the whole thing stops. So the good thing about this is that the non-functional alleles are recessive to the functional dominant allele, meaning that it does you need to actually have both alleles to give you an inactive enzyme. So for example, here I'll give you this white. You have basically, as you can see here, double AA. So this is basically it's giving you an enzyme. So enzyme A. This BB is not giving you enzyme B because both this is basically is homozygous. So you have you don't have a good copy of the B protein. So that's why you have white because basically you need both of them. So same thing here. This is the opposite. Now here you don't have the A what's it called enzyme, but you have the B enzyme. But here you have a copy of actually one good A enzyme and one copy of the B enzyme. So this actually can give you the whole purple color because now, as you can see, you have both enzymes working functionally. So now when you basically let this self cross, this is what you get. These are all the different combinations of alleles you get after segregation, and you do the Punnett squared. So you get basically, as we said, this nine purple and seven white. So let's give an example here. This is actually purple because it has one A and one big B. So it has actually the enzyme for basically gene A and the enzyme from gene B. But for example, here, this guy over here, it has the enzyme for A, but it doesn't have it for B. Same thing goes maybe, for example, here, you have the enzyme for A, but so you don't have the enzyme for A, but you have the enzyme for B. This one as well, you don't have the enzyme for A, but you have the enzyme for B. This guy over here, you have the enzyme for A and B. Same thing, all these purple ones actually have, a good functional allele for the basically, gene A and gene B. So for example, let's show this here. So you have this is the precursor, this is the colorless pigment. So the first has to go through, this intermediate colorless, which is done by enzyme A, and then this goes to purple through enzyme B. So you need functional enzyme A and enzyme B, both of them, for this precursor to become a purple pigment. You need them. So if any of them are not there, forget about it. And that's how two genes that encode for proteins that participate in a sequence in the biochemical pathway can give you epistasis. Because here, these two genes, so the gene basically A and gene B give you, enzymes A and B that are working in the same biochemical pathway. and how these can affect each other. Because if this guy is not there, it's not affecting directly enzyme A, but it's affecting the whole, basically, producing this pigment. So you're changing again the phenotype. Now I'm going to give you another example how actually products of one gene can mask the expression of another gene. And this is quite cool because you see this a lot in Labrador dogs. So Labrador dogs basically have a pigment. And either this pigment can be black, which is actually quite dominant, and they actually have a pigment that is brown. So we see Labradors that are either black or we call them basically actually, chocolate Labradors because they're actually brown. And they have basically the recessive gene, which is basically this lower, this small b. So these guys actually show a complete dominance inheritance pattern. So similar to Seeger and with Mendel's. Either these guys have the B, the big B, so they're black, or they have the small b, which is brown. If they have basically a copy of this black and this brown, they're black because it's complete dominance. However, so these genes are giving you the color of the pigment. This encodes for the pigment color. If it's either black or brown. However, there's another protein that's gonna basically help it deposit in the hair, and that's called the E gene. So if the E gene is actually there, so this is dominant. If it's functional, it's dominant. If it's there, it's gonna actually put either color pigment in the hair. However, if this is not actually functional, it's not gonna put the pigment in the hair. It's not going to deposit it. So you could actually have either black or fully brown. But if this allele is not there, well, those are going to be produced, but they're not going to be deposited in the hair. So the dog actually will not have any pigments in the hair, and this is what gives you actually the yellow color. It's the absence of these basically guys. So let's put this in an example. We get this heterozygous black labs. So basically, it has this big B, small b. So this guy is, actually is a black Labrador, and basically, it has this E. So this is the receptor that puts the melanin in the hair. So we see that it actually has a functional one. So it's a black Labrador because it's producing black pigment and it's being deposited in the hair. So you might expect this ratio of this 9331. This is the one that, again, same, what's his name, Mendel's, when he was testing two traits, was giving. But you don't get that. You get this 934. 9 black, 3 brown, and 4 yellow, which has no pigment in the hair. So we cross these two. So these basically be E, and this is what you get in the Punnett square. So this guy, for example, let's go one by one. So this guy over here, we know that has the black pigment, and it actually has the receptor that's putting it in the hair. Great. Same thing for all of these guys, but this guy, for example, here. This guy has actually the brown, because it's all small b's, because these are recessive, so it has the brown pigment and is being deposited in the hair. Great. But now if you take a look at this guy has the black pigment, but it doesn't have the receptor that's putting that pigment in the hair. So that's why this guy is actually, um, yeah, you're basically blonde. So this is why we have actually blonde Labradors. Same thing with here. As you can see that this receptor, if this receptor is not there, it's automatically this guy is blonde because it's not going to deposit in the hair, so the hair is not going to get any pigments. No matter what pigments you have, doesn't matter. It's not going to be put in the hair. And for example, this guy over here has the brown pigment, and it actually can be put in the hair. So that's why you get actually um, the basically brownness. So we see that basically this gene can actually mask. For example, this guy masks, for example, the black pigment. This actually masks the brown pigment because it's not there. It's not gonna basically put it in the what's it called, the color and the hair. So you're not gonna get the brown in this case or the black in this case. This is how these things can be masked. So it's just to show you that Mendel actually had a very good way of looking at things, but they were very naive because of all of these seven different things that can occur. Thank you so much, guys, and hope this has been clear. If not, please ask me anything you want.